Thank you very much. Thanks everybody for coming to the session. There's our forward-looking statement. So, my name is Willie Mesa. I'm one of the co-founders of Athersys and the Executive Vice President of Regenerative Medicine. And I'm going to spend a little time this afternoon talking about our platform technology, MultiStem, that we're developing for treatment of critical care injuries and diseases like ischemic stroke, which are a substantial unmet medical need in today's world. 2022 has been a year of refocusing and reinvigorating the organization. We have a new management team that's come in, including a new CEO, Dan Camardo, who's here in the back of the room, that brings new expertise in the space of clinical to commercial translation. Our lead product is called MultiStem, and I'll be talking more about that in a little bit, but it's an allogeneic bone marrow-derived adherent cell type that we're developing for the acute care market. And we've made a lot of progress in the manufacturing of the cells, learning how to make hundreds of thousands of doses from a single 100 mil bone marrow donation, and we're now in a 3D manufacturing process. So we acquired the rights to these cells almost 20 years ago now, and I've been involved in trying to figure out what the cells do, and more importantly, what the cells don't do for almost that entire 20 years. And normally when I give these presentations, or scientific talks about the cells, we would go through in painful detail all of the things the cells do, all of the indications that we worked on, all of the cool mechanisms of action that the cells participate in, the different animal species, the different academic researchers we've worked with, how they modulate the immune system, how the spleen is an important organ. We've got more than 40 peer-reviewed articles over the course of the last 20 years demonstrating what the cells do and what the cells don't do, as I said, and we've treated hundreds of patients. But I've been told by some of the people in our group at Athersys and by some friends that, you can't drink a lot of water out of a fire hose, and so maybe we should tone it down a little bit and try to give you less information and do a deeper dive on maybe just one or two interesting things about our company and about our product. So MAPCs, multipotent adult progenitor cells, as they were originally named when they were characterized by Dr. Catherine Verfey back in 2001, we call them MultiStem. That's the clinical grade product. So if you hear me say MAPC or MultiStem, those are equivalent throughout the remainder of the talk. They're derived from bone marrow. We didn't publish papers, but Catherine originally published papers showing that the cells could be isolated from multiple different tissues, and they're a distinctive cell type. We get grouped in, we're told frequently that we're MSCs or we've renamed MSCs, and I'm going to show you some data that would support that, in fact, these are not MSCs. They are a distinctive cell type. So one of the things we've done, and we've done a lot of, is we get bone marrow aspirates from willing donors. And on the right-hand side here, you can see this is a bone marrow donation from a single donor that we split in half, and we grew the cells up under either MSC conditions or under MAPC conditions. And you can see the preferential growth and expansion benefits in the MAPC population compared to the MSCs. They have different sizes, the MAPCs, and I'll talk more about this in a second, but the MAPCs are much smaller and less stromal than the MSC population. And when we do principal component analysis of the entire gene expression profile of the cells, MAPCs and or multistem cells clearly segregate away from MSCs. Paper published by a group that we didn't even know existed or had any kind of connection to at the time they published the paper, demonstrated that the two cell types have very different size. And when you label the cells and you give them intravenously, that the distribution of the cells is very different. So you can see that the MSCs end up in the lung, and you see some of the MAPCs in the lung, but you also see some of the MAPCs in the spleen. And there's some reasons for this other than size, and I'll show you in just a second. We've done a lot of experiments, again, as I said, where we've isolated cells or bone marrow from one donor, and then we've split it, and we've gone on to do more complex analyses. So here you can see a couple of different MSC lines, a couple of different multi-stem cell lines, and you see that within each population of cells, there's roughly a 99% correlation internally to that cell type, but there's only an 85% correlation between the two different cell types in this experiment that we did. So, we're 98% identical to chimpanzees, but the 2% makes all the difference in the world. So here we got about a 15% difference in this transcript analysis. We went on to take some of the interesting genes and saw things that were up in multi-stem that were down in MAPCs, and things that were down in MAPCs that were things that were up in multi-stem and that were down in MSCs, or things that were up in MSCs that were down in multi-stem. Got some antibodies, did some immunofluorescence staining, and lo and behold, you can see how these two different proteins are clearly present in one cell type but not in the other. And we also did some analysis of secreted factors. So you can see this is a standard commercially available angiogenic factor immunoblot. And you can see here there's 3 donors on this data set. Donor 147, donor 149, and donor 153. So in the top, they're MSCs isolated from these donors, and at the bottom, they're MAPCs isolated from the donors. And you can see when you load the same amount of protein into these immunoblots, how different the secreted protein profiles are. So we see gene differences, we see protein differences, we also see functional differences. So here, we're looking at human aortic endothelial cells. So they're a standard type of endothelial cell used in looking at binding assays, looking at rolling and extravasation of immune cells. So we're looking at 3 markers here, E-selectin, VCAM, and ICAM. These are 3 markers that are upregulated during pro-inflammatory signaling on endothelial cells. So you can see over here that each of these proteins is at a very low level before you basically license them with TNF-alpha. And so we set this bar to 100, and you can see that the MAPCs have a dose-dependent effect on being able to drive down the expression of these different proteins on the cell surface of the endothelial cells compared to MSCs, which don't seem to have much of an effect at all. And this leads to an actual meaningful benefit when it comes to the binding of neutrophils to the surface. So you can see here, if you have unactivated endothelial cells, there's not a lot of neutrophils binding. They go up significantly when you license them with TNF-alpha. The MAPCs, or the multi-stem cells, knock this binding pattern down, as opposed to MSCs, which have virtually no effect. So we're able to demonstrate gene to protein to actual functional difference between these 2 cell types. So this is kind of a very busy slide, but I like it when I'm talking about the specificity of how the cells are working in acute neurologic injuries. But what we believe is that if you can intervene early after you have an acute injury, be it a TBI, stroke, spinal cord injury, hemorrhagic stroke, that you can change the immune cell infiltration into and across the blood-brain barrier into the brain, have less microglial activation, have less pro-inflammatory cytokine secretion, and change the profile for how the cells are actually working to lower the threshold of pro-inflammatory signaling and drive up the reparative aspects of the immune system. So we believe that the cells work agnostically against, irregardless of what the injury is, that they stop or mitigate the pro-inflammatory cascade, and if you get them into the body in the right time frame, they'll rebalance down pro-inflammation and up with reparative aspects of the peripheral immune system. So with that as kind of our working hypothesis for this specific mechanism of action, we've gone after and turned our attention to translational research and ultimately clinical research across the number of acute indications. So we're currently in a pivotal phase 3 stroke trial called MASTERS-2. We also have an ongoing phase 2 trauma trial. This is resuscitation from hemorrhagic trauma that's being conducted. It's funded by the Department of Defense. We've also run other trials, acute respiratory distress syndrome, or ARDS, things like graft versus host disease, acute myocardial infarction. We've run previous trials in those indications, but we have a whole portfolio of other indications, including applications in the animal health space, utilizing these cells to treat a variety of indications. So previously, as an organization, we were looking to have a successful stroke trial and not talk about or contemplate being able to partner or find people that might be interested in some of these applications. Well, we're not that organization anymore. If someone has their favorite indication and they want to come talk to us, I'll try to be as obvious at the reception tonight as possible, and come find me and we can talk. Just to highlight again some of the aspects of that little cartoon I showed you a little bit ago, here's two different acute injury model trials. We have MASTERS-1, our phase 2 acute ischemic stroke trial, and MUSTARDS, which was our acute respiratory distress syndrome trial. And you can see the same effect on the same pro-inflammatory cytokines. Two different target organs, two different injury models, but the same effect of the cells on lowering the inflammatory burden in these patients. This is another important aspect of what we believe about ourselves, and that is most acute ischemic stroke trials have a 90-day endpoint, because the greater majority of all therapies that are tested in acute stroke, what you are at day 90 is what you're going to be for all intents and purposes for the rest of your life. You can see there's a difference between the multi-stem treated and the placebo-treated patients in our phase 2 stroke trial. It wasn't statistically significant, but we had seen in multiple animal models that the longer you kept animals on study after treating with multi-stem compared to placebo, the more differences you saw in the cell-treated animals. And so we pre-specified this one-year endpoint, and we see a statistical significantly different delta of roughly 21%. And this is the kind of data that led the FDA to give us RMAT designation, fast track designation, and special protocol assessment for our program. And most recently, our partner in Japan, Helios, had a readout on their phase 2/3 acute ischemic stroke trial called TREASURE. And basically, this is kind of busy. I'm not going to spend a lot of time explaining it, but this is called the modified Rankin. This is a standard outcome measure in the stroke community for looking at people across the entire spectrum of injury. So an MRS of 0 is totally normal. There is no sort of deficit. 6 is death. So what you want to see is you want to see patients progressing towards the left-hand side. And so you can see at day 90, there's differences between the cell-treated and the placebo-treated, but it's not statistically significant. But at 1 year, the shift is more dramatic, and more patients are moving towards the recovery end of the spectrum. So we've taken these cells, we've gone from being able to understand how to formulate the cells into vials, how to make it convenient to use in the acute care setting. We're to the point now where it's just thaw and administer, but I want to end with one last breakthrough we've kind of made as a company, and that is upstream of this. Storing cells at an ultra-low temperature is problematic in pharmacies and in hospitals. So we were hearing this feedback from all of our stroke neurology groups about how they only had access. The cell therapy lab was only open 9 to 5 because that's where they had the liquid nitrogen. So we basically came up with, engineered, and now have intellectual property around something called SIFU, our Secure Integrated Freezer Unit. So this is a hospital and pharmacy-friendly solution to storing cells at ultra-low temperatures at the hospitals. You don't need liquid nitrogen, plugs into a standard power outlet, makes the cells 24/7 available. All you need is the code to type in. It's basically an ATM for cells as opposed to cash. And so we currently have prototypes available that have met all of the specs to date. We've built some flexibility into the CFU, and we're currently seeking partners to potentially co-develop and or license this for end-chain users in the cell and gene therapy space. So with that, we've really spent a lot of time the last 20 years trying to understand from isolation of the cells, growth of the cells, to the patient, storing of the cells, how to make things as easy as possible for making allogeneic cell therapies more attainable in the near term in the clinical setting. So with that, there's our summary, and I thank you for your attention.